I'm Ryan Schaffner. I will be speaking about true bugs as a target group for assessing the impact of the New South Wales 2019 and 2020 wildfires. My co-authors are Marina Chang and Jerry Cassis. We thank our co-contributors to this project, Sean Laffin, Chris Reed, Bill Sherwin, and Wendy Shaw. We sincerely thank the New South Wales National Park and Wildlife Service for granting us permission to conduct this research on national park properties, and for their invaluable assistance in accessing difficult locations. Assessing the conservation of invertebrates in Australia is a major challenge. There is a massive taxonomic impediment due to numerous undescribed species, as well as sampling inadequacy relative to regions like North America and Europe, or even other taxonomic groups like mammals and birds. This is despite the fact that 75% of all living eukaryotes are invertebrates. Many invertebrate species are narrowly distributed, which places them at risk of extinction due to landscape-scale disturbances such as wildfires. With the forests of the Great Dividing Range of eastern Australia a known hotbed of organismal diversification, we hypothesized that this region represents a heightened invertebrate extinction threat, especially with events like the 2019 and 2020 wildfires, which were massive in both scale and severity. We chose true bugs as a target group to assess the impact of the 2019 and 2020 bushfires on the invertebrate fauna of northeast New South Wales. True bugs account for approximately 3% of the described metazoan diversity, or, in other words, they account for 3% of all described animals. True bugs are hemimetabolous insects, which means they undergo incomplete metamorphosis. There is no pupal stage like there is for insects like butterflies, beetles, or wasps. True bug mouthparts are unique among the insects. In true bugs, the mouthparts have fused into a hollow piercing, sucking tube. True bugs occur across a wide range of habitats, both aquatic and terrestrial. They can be predatory or phytophagous, which means to feed on plants. Many of these phytophagous taxa are host-specific. They only feed on a few species of plants, and these plants are often closely related taxa. This close association with their host plants increases the susceptibility of bugs to environmental disturbance, as any disturbance that affects the host plants also affects the bugs. Our goal for this project is to evaluate whether any of our target taxa warrant protected status under governmental and/or IUCN criteria. High-priority, at-risk species will be nominated to the Threatened Species Scientific Committee for listing based on several factors: the area of occupancy, the extent of occupancy, measured genetic diversity, and population size based on the number of individuals collected. To determine which true bug taxa we would target for this project, we examined historical collection data for true bugs in Australia. This data was sourced from the Plant Bug Inventory, which hosts museum collection records from around the world. We also examined specimens from the UNSW Insect Collection, which is comprised primarily of true bugs from Australia. We restricted our search to only those taxa that occur within the northeast forest region of New South Wales. We then selected our targets from among these taxa based on a combination of two factors. First, we looked at how geographically restricted the range is for each taxon. Several of our targets are known only from within the Northeast Forest region. Second, we determined what percentage of the historic records occurred within areas burnt by the 2019 and 2020 wildfires. From this, we identified 9 primary target taxa, all of which had over 75% of the historic records occurring within burnt areas. We also identified an additional 30 taxa of potential interest that did not quite meet our criteria, but could be the subject of future research. To conduct our survey, we revisited as many of the historic collection sites for our target taxa as was possible. We specifically targeted the known host plants for each target taxon at each site. We used a beating stick to dislodge bugs from vegetation. The bugs fall into a beating sheet held under the vegetation, whereupon they were captured using an aspirator. At each location, we attempted to collect off of all known target taxa host plants that were present. We also collected off of non-target plants as an additional measure to collect our target true bugs. In addition to the historic sites, We also used host plant distribution records to determine new survey sites for our target taxa. During our survey, we sampled both unburnt sites and sites that were burnt in the 2019 and 2020 wildfires. As part of our investigation of the impact of the wildfires on invertebrate taxa, we are examining whether bushfires act as a barrier to dispersal for invertebrate species. To determine this, we are working in conjunction with Australian Museum to examine the population genetic structure of two invertebrate species, Epimixia dismica, a true bug, and Amphistomus primonactus, a flightless dung beetle. The overarching question to this part of the study is, how related is one population to another? We can calculate this by examining single nucleotide polymorphisms, which are single DNA base pairs that differ from a reference sequence. By examining the variation in single nucleotide polymorphisms within and between populations using various genetic differentiation methods, such as FST or Shannon information, we can determine how related one population is to another, and by examining a matrix of these comparisons, we can determine how much dispersal there is between populations. To determine whether the bushfires have had an impact on this dispersal, we collected samples from populations that were separated by burnt areas, but we also collected samples from populations that were not separated by bushfires. By using pairwise comparisons, we can hopefully determine whether these fires have had an effect. We are currently in the thick of things with our data analysis, but we do have some very preliminary results. Overall, we collected 159 morphospecies of true bugs. However, we were only able to collect one of our 9 primary target taxa, which is a plant bug of the genus Woodwardiola. The bug in this picture on this slide is one of these Woodwardiola, and this picture represents the first known photograph of a live specimen. We were able to find Woodwardiola from many sites in the region, with almost 200 individuals collected. As common as this species was, however, we were only able to collect them from unburnt sites, even though their host plant, Lomandra longifolia, was numerous in recovering burnt areas. We collected very few specimens, and a commensurately lower species diversity, from sites that were burnt in the 2019 and 2020 wildfires. Burnt sites had far fewer host plants, both in terms of individuals and taxa. From the host plants that were present in burnt areas, we saw very little evidence of recolonization by true bugs, as evidenced by Woodwardiola. We ran into many difficulties with this project, some of which have no doubt influenced our dataset. One of our biggest issues has been COVID-19, which delayed the onset of fieldwork by several months. This in turn meant that the seasonality of many true bug species, which often exhibit higher abundances in spring, might have impacted the number of taxa we were able to collect during the summer. In addition, March saw record rainfall in this region of New South Wales, which prevented our access to many field sites due to blocked roads or other unsafe conditions. This was coupled with general inaccessibility of historic sites from a wide variety of factors like remoteness. And as mentioned previously, burnt sites were difficult to collect in, as many times there were no plants within reach for us to collect off of. We were also limited to collecting sites that were managed by New South Wales National Parks. The permitting situation was such that we could not expand our scope within our required time frame. Due to the limited number of historic records of true bugs, coupled with the difficulty in verifying the presence of populations for a given taxon of true bug, traditional species risk assessment methods, like area of occupancy and extent of occupancy, are problematic when assessing invertebrate taxa. In order to improve assessments of potentially threatened invertebrate taxa, different strategies are required to address the shortfall in occupancy data. Long-term monitoring strategies should be investigated as a mechanism for evaluating at-risk invertebrate taxa. As the frequency and intensity of events, like the 2019 and 2020 wildfires, increases due to climate change and other anthropogenic influences on the planet, we will need bold new strategies to ensure that our biodiversity is preserved for future generations.